Hello and welcome back everyone to week 6 of the YSP 5th grade spring curriculum. This week we'll be covering the lesson on chromatography and leaf pigments. I just want to say that this is probably one of my favorite lessons that I've taught across both semesters, fall and spring, just because a lab process that was considered to be pretty complicated in an actual chemistry lab can be replicated in the 5th grade classroom using easy to access materials. And I found that really cool, and your students are going to find it pretty cool as well. Looking at the materials, the tall transparent cups have my attention first. These should be similar to the ones that used in the photosynthesis lesson. Hopefully they're going to be around 5 to 6 inches tall, enough to hold just short enough to hold an entire piece of chromatography paper standing in lengthwise. It should be around that tall because the chromatography paper will be suspended, hanging inside the cup, not touching the bottom or any of the sides. Make sure you have enough isopropyl alcohol. That's just rubbing alcohol, so if you are short and the JEP office is closed, it's pretty easy to go buy that on your own, but if you check beforehand, you should be able to access the JEP house and the nucleus and get any other supplies you're going to need for this lesson. If you're unfamiliar with chromatography, I'll just go over a pretty basic summary of it. It's when you use rubbing alcohol that travels up a piece of chromatography paper, and there's a little bit of some sort of dye on the chromatography paper. And as the rubbing alcohol travels up using capillary action, then the different dyes in that certain spot of color on the paper start to split. And in this lesson in particular, we're going to be splitting the dyes that are present in a leaf. And those might show up to be like orange or yellow or green, because those are the colors associated with leaf growth. Again, chlorophyll is a big part of this lesson because we've been talking about over the past couple weeks plant cells, photosynthesis, and trying to figure out how plants get their energy, and this lesson goes even further into that subject, specifically focusing on what makes leaves turn green and change color through the seasons. Now, moving on to engagement, the students might not necessarily be as familiar with the concept of leaves changing color. They probably will be, but they might not have experienced it to the extent of people from other areas of the United States have been able to. So including some pretty pictures or really cool examples of what forests or entire areas look like when they're orange and red and brown is pretty cool, and my students found those very cool to see. As for the question of the day, what makes leaves change their color, there's a lot of different things that the students can bring into play. Temperature gets colder in the winter when leaves change their color. Some of them might even be able to bring up the idea that the days get shorter in the winter, meaning that the leaves get less sunlight. That could tie back into when we did the lesson way back in the fall about planetary body movement. But there should be a handful of explanations for why the leaves change their color, and this can prompt some good class discussion. Going on to the next question about separating different sized particles, that's just another way to get their minds to grasp onto what the chromatography paper is actually doing in separating the different pigments. There's some other ideas that I might go over later in the video. If some of these ideas, as highlighted here, aren't necessarily assimilating with the students' minds as easy as they should be. To be adequately prepared to present this lesson in front of the class, I do recommend you watch a couple of videos on YouTube. There's a ton of people who do this experiment just so you can figure out what works best for you and then test it a couple times yourself so you know exactly what needs to be done so your students can get similar adequate results than what you get. One of the big things that I want to tell you to do is to have the students pick green leaves. And it's important, like, the lesson would be cool if we could try different colors of leaves, but green works the best for a situation like this because those leaves haven't dried out yet. The dye from the leaves is still easily transferable to the chromatography paper, causing it to separate into the different colors as we want it to. As for acquiring the leaves, I know some TAs have brought in their own set of leaves to save time at the beginning of the lesson, but in my classes I found it more enjoyable to let the students out into whatever outdoor area they have at their elementary school and pick out some leaves for themselves and then decide as a group which leaves that they want to do for the experiment. And you can also choose one volunteer to have their leaf be chosen for you to do your part of the experiment. That helps them feel connected and immersed in what they're doing for today's science lesson. If they feel like they contributed to it, brought in some of their own materials, or picked the leaf that they're going to be working with. And just to emphasize that last point, you should be doing this experiment along with them. I found it very helpful. My class had a document projector, so I could take into account using a pencil to draw the lines under the document projector to make it really easy for the students to follow and do all the same measurements I was doing, as well as where to place the leaf and where to roll the coin along the line to make sure that the students knew exactly what to do by using what they saw on the front of the board on the projector as a model. I'll put up a picture about what the final product should look like to go through steps 4, 5, and 6, so you know where to put the popsicle stick, where to tape it, where the chromatography paper should be positioned. Again, if you still are unsure, try to find some YouTube videos or talk to some other TAs to figure out how to approach that. Looking at bullet point number 7, the waiting 10 to 30 minutes for the alcohol to travel up the paper. This could end up taking the full 30 minutes, which is why I recommend you start doing this lab or start doing this experiment as early as you can within the hour or hour and a half of your lesson. That means you can take this time to go over some of the more advanced or the more complicated topics that are specifically related to chromatography that we'll talk about later, so we can adequately use that time to get as much knowledge and talk about science for as long as possible with the students, because we don't want them to just be sitting and waiting, watching very, very slow movement of alcohol up the chromatography paper. I mentioned the elaboration section, and this is something that I do want to go over because there is the potential for this to get kind of confusing for the students. The difference between chlorophyll and carotenoids, I'm pretty sure that's how it's pronounced, you can check that for yourself, allows them to see the difference between the green pigment and the yellow pigment when they separate the pigments through the experiment, and those are the two that are going to be most prominent on their chromatography paper, those two colors. I mentioned earlier in the video that we'd be going over other ways for them to understand which colors travel faster or which ones travel farther, and the fact that this has to do with size. There are a couple animations for what chromatography should look like with smaller particles moving faster or how particles are being separated that you could find and download to put in or play on the screen during your lesson. But some analogies that I've heard, there's the guinea pig and hamster one that's up near the top of the page. The one that I did in my class is, if you think of a ping pong ball and a basketball, they're obviously very different in size, and you just let them be blown in the wind. The ping pong ball is going to move a lot faster by the wind, and the basketball is going to be stuck because it has more mass and it takes more effort for the wind to push it and make it travel a longer distance. The last couple things I wanted to cover, going into the evaluation section, the whole idea of a tree species, I don't know if this will stay in the lesson moving into next year because there's there's very little way of knowing the species of the tree that the students are going to be working with, and this lesson is more focused on separating the pigments and figuring out how that process works or how leaves change their color in the first place, rather than figuring out what specific types of trees create which specific pigments, because there's not much variety that the students can pick with around their elementary school campuses. And with that, by the end of the lesson, the students should be able to answer the question, why do leaves change color and how can we measure it? That new added part, how can we measure it, that wasn't there at the beginning of our discussion on this lesson, is the process of chromatography, which we can use to separate the different colors within a leaf to kind of analyze how they change color. And with that, that's going to be it for week 6, the lesson on chromatography.